Hi, everyone. I'll just make sure this is adjusted because I know I have a quiet voice. Some people say they can't hear me. So if you can't, and everyone hear me? All right? Yeah, barely? Okay. Oh, maybe that. There we go. Is that better? Okay. Won't be able to see my speaker notes, but that's all right. So pleased to be able to chat with you today about this project that we worked on with some of the irrigation districts and the University of Calgary. I'll just acknowledge, it's up on the screen there, but the districts that partnered with us on this were the Bow River Irrigation District, Lethbridge Northern, Raymond, and the St. Mary Irrigation District. There we go. As we all know, irrigation infrastructure is designed to convey water, and often the ability to convey this water is challenged by aquatic growth in the irrigation canals. So irrigated crop production really depends on timely receipt, timely delivery of water at critical times in crop growth. So when you have weeds that are slowing down the flow or plugging filters or screens, it's a big problem and can really affect crop growth and production. So because of this, the irrigation districts spend a lot of labor on physical removal of the weeds, and also they spend a lot of money on the chemical treatment. So vegetative growth is caused by a lot of different factors in the canals, including temperature, light penetration, but also we know one of the main reasons we have excessive aquatic growth is because we have too many nutrients in our canals. So if we could figure out where those nutrients are coming from and what's happening to them as they flow along the canals, then the irrigation districts can use this information to target their nutrient management practices to hopefully reduce the nutrients, which would then reduce the amount of weeds that are growing. So an example of this would be, and this is a bit of a spoiler alert, but if you're finding that most of your nutrients in your canals are, say, coming from soil, then as a district, if you targeted things that would minimize soil erosion in your district, then it's likely that you would reduce the nutrients in your canals. That's the idea of this project. So these are the sources and sinks that we targeted for this project. We just kind of took an educated guess at what we thought may be some of the probable sources and sinks. So the things that we investigated for this project were, as the sources, were manure, soil, some urban wastewater, fertilizer, drainage water, and atmospheric deposition. Then on the flip side, the sinks, which is basically areas where nutrients could accumulate and then perhaps be released at a later time, were like sediment in the bottom of the canals, aquatic plants, canal water, and also periphyton. So as far as the aquatic plants go, we targeted the plants that were growing along the edges of the canals that likely had their roots down kind of into the saturated zone of the canal. And periphyton is algae, so it's just what's growing and accumulating on the rocks and stuff in the districts. So when it comes to the fate, what we did was we just kind of followed the nutrients as they flowed downstream in the canals. So we were looking as to whether there was spots where we saw there was a definite input of nutrients, or maybe a spot where the aquatic plants were taking up the nutrients, or maybe there was deposition obviously happening into the sediments, or whether there was just chemical transformations like denitrification or something that was happening within the canals. So now, I know you're all super anxious to find out how on earth we did this nutrient source tracking. So let me introduce you to stable isotope analysis. So most of us are really familiar, or maybe somewhat familiar, with single atoms of elements like nitrogen or carbon or oxygen, but we might be less familiar with the term isotope. So let me take you back to your high school chemistry, see if I can kind of explain. So isotopes are atoms of the same element that have different mass numbers. So they have the same number of protons, but they have different numbers of neutrons. So for example, nitrogen, all nitrogen isotopes have 7 protons, but some nitrogen isotopes have 7 neutrons and some have 8 neutrons. So the mass, you get the mass number by adding the neutrons and the protons. So some nitrogen isotopes will have 7 protons with 7 neutrons, so 14, and some will have 7 protons plus 8 neutrons, which is 15. So that leads us to the names of these nitrogen isotopes as 14N and 15N. So what else about? Yeah, oh well, one of the other isotopes that we commonly hear in the news is carbon-14. So that's an isotope that they use for carbon dating and that type of thing. So the other thing too is, well, we're all really familiar with how we might measure single elements in water, like nitrogen. We measure it as a concentration, as in milligrams per liter in a water sample. When we're measuring isotopes, it's different. They actually are measured by the ratios of the different isotopes. So we can use that kind of as a benchmark, because the different ratios, or different sources, different natural or anthropogenic effects will affect the different ratios of the isotopes. So that can help us to kind of track where these are coming from. So I hope you're still with me, because that's pretty much all I know about isotopes. So that leads me to put in a really big plug for our partner at the Isotope Science Lab at the University of Calgary. They did all the isotope analysis for us, plus they were really instrumental in helping us to analyze the isotope data that was being produced from this analysis, helping us to interpret it and apply it to what that actually means in terms of the nutrients that were in the canals. So moving into the methods, this project was a 2-year project, 2019 and 2020. As I said, we had 4 participating irrigation districts. So each of those districts selected 3 irrigation canals. They were asked to target canals that maybe had water quality concerns or had weed problems or stuff like that, so that we could really kind of hedge our bets in basically getting data that we would really be able to use this isotope data to maybe figure out some problems. So, and then within each canal, there was 4 sampling sites. So these sampling sites were located about 3.5 kilometers apart from each other. So, and these were located, I better read this so that I get it right. The 3.6 kilometers represents 5 times the average uptake length of nitrate N. So this nutrient uptake, which is also called nutrient spiraling, refers to the process in nitrate cycling that occurs in moving water systems. So basically, the uptake length is the average distance traveled by a nutrient molecule in inorganic phase before uptake. So we just kind of increase that up to say, okay, let's go 5 times that length just to make sure we're allowing for adequate distance for this cycling that may or may not be happening, but we just wanted to make sure we were allowing enough distance, so if that was happening, we would be able to capture it. So canal water was sampled in July and in August, so twice a year for 2 years. The sources and sink sampling were collected once a year, and analysis of these samples was done for both concentrations, so the actual nutrient values, and also the isotopic ratios. So, okay, So this graphic will be kind of familiar. You would have seen it before. These are the nutrient sources and the sinks that we tested. So I'll just move this over to the side. I just kind of want to show you some pictures of what we were actually sampling so you can kind of frame it in your brain a little bit. So as far as the manure goes, manure was collected in liquid form from some dairy barns. Solid manure was collected from some feedlot pens, and feedlot catch basins were also sampled. Then moving into the soils, so we sampled 4 different types of soils. So we sampled manured fields, unmanured fields, but that had been treated with inorganic fertilizer. We sampled some fields that had recently just grown nitrogen-fixing crops. And then we also sampled grasslands or pastures, I guess kind of a little bit like a control, right? So something that has not really had any nitrogen added to it, either from fertilizer or from manure. Yeah, and then one of the irrigation districts had a canal that was used in the off-season for a licensed delivery of treated wastewater to the river. So because they were wondering whether that use may have some effect on the nutrients in that canal, we added in some municipal wastewater. So we sampled 3 different municipal wastewater treatment plants, and they're treated effluent to see if that was the source of some of the nutrients that we were seeing. And yeah, and then we also, one of the most interesting things that I think that we sampled was drainage water. So because drainage water often, it's not supposed to, but it often does make its way into irrigation canals. So we sampled both the tile drainage samples and also surface runoff. So we did this both in the spring for spring runoff and also during a precipitation event. So then on the flip side, as far as the sinks go, we took some samples from the sediment at the bottom of the canal. We did that at the end of the year, obviously when the irrigation water is turned off and the canals had been let to dry out a little bit. We sampled the sediment. Also sampled the aquatic plants, like I said earlier, the grasses, the reeds and stuff from along the edges of the canals, but also any floating aquatic plants that we might have found. We sampled the canal water, and also we took some algae from the rocks that were submerged for the periphyton samples. So I've alluded to a lot of these objectives already, so I'll just quickly go through them. So there was 4 objectives for the project. So to quantify the longitudinal changes in nutrients in the irrigation canals, investigate the source and fate of nutrients within the canals, to use this nutrient source data to inform management decisions, and also just to evaluate this technology and see whether it's applicable to the irrigation districts. Is this a tool that they can use to help their nutrient management programs? So moving into some of the results. So as far as the first objective, which was to quantify longitudinal changes of nutrients in the irrigation canals, I'm sure this graph is likely a little bit small for you guys at the back, but I'll just kind of step you through it. That basically, from top to bottom, those are the 4 different irrigation districts. So gosh, I can barely even see it from up here. I think BRID is at the top, LNID, maybe you can read. Anyways, it's all the irrigation districts from top to bottom. And then the 3 columns from left to right represent the 3 different canals of each of the districts. Then there'll be kind of usually 4 dots within the boxes. So those kind of represent the 4 sampling locations, upstream to downstream in the canals. And then the different colors represent the 4 different sampling events. So there's a lot of information there. Generally what the result, even though we occasionally see some sites during some sampling events are different than the other ones or might spike up. Generally, when you look overall, nutrient values in the individual canals were very low. They were pretty uniform. There was no obvious consistent nutrient inputs or transformations that occurred along the studied canal stretches. So moving into objective number 2, which is where we get into the actual isotope stuff. So this graph here, we're going to first talk about it in terms of what we're seeing in the canal water samples. So as far as carbon goes, it was a pretty easy, straightforward answer across all the districts, all the canals, all the sites, all the sampling events. All the carbon was all from erosion of carbonate rocks. So I say it's kind of a cut and dried answer. There's not really a lot we can do about geological weathering of rocks. So as far as phosphorus, it's also kind of a short story. What we actually found was that the concentrations of phosphorus in the irrigation water were too low for isotopic analysis. We actually knew that, we were warned that that was likely the case, but the U of C was interested in still trying. They kind of just wanted to see how far they could push their technology to see if they could actually get some results. So the first year of the project, they did attempt to analyze for phosphorus, but they were unsuccessful. So basically phosphorus concentrations in our irrigation water, we can't use stable isotope analysis to track their sources. But when we get into the nitrogen, this is where things get interesting. So that's what the graph there is showing. So across the bottom, that's the concentration of nitrate in, well, I should say, first of all, it's a graph of all the canal water samples that were above the detection limits. So the bottom, the x-axis, is the concentrations of nitrate in the canal water, and the y-axis are basically the ratios of 15N. And if you look at the symbols, I don't know if you can see the symbols from back there or not, but the circles represent BRID, the triangles are LNID, the squares are RID, and the crosses are SMRID. So what we saw was that there was basically 4 groups that kind of emerged from this data. So the first group I'll talk about is group number 1 that's circled in red there. So this was a group, it was basically the nitrate in the canal water could be aligned with the nitrate that we were finding in soil, basically soil-derived nitrate. So the nitrate that we were seeing in the soils that were coming from the N-fixing crops or the grasslands. So not a lot of, or if any, manure influence on those soils. So basically soil-derived. So what we see is, what the percentages listed there are that 92% of SMRID's canal water was associated with this, or I should say 92% of the nitrate 92% of SMRID's canals, the nitrate that was in the canals was associated with this soil-derived nitrate. And then going down, 3/4 of BRID samples were associated with this soil nitrate, half of LNIDs, and about 1/4 of RIDs. So now if you're following me, and it always kind of catches me up too when I look at that, I'm like, it doesn't add up to 100, but that's okay, because I'll just remind you that it's actually, once we get all the percentages for SMRID, it's all the SMRID percentages that are going to add up to 100%, and so on and so forth. So moving on to group number 2, which is associated with soil-derived nitrate that was from soil that had repeated manure applications. So we see about 1/4 of LNID's canal samples were associated with this, 12% of BRID, 8% of SMRID, and only 5% of RID. Up to group number 3, this is that the nitrate in these canal samples were associated directly with manure. So we see that most of RID samples, so 69%, were associated directly with manure, 20% of LNID, and 12% of RID. So, and there was no SMRID samples in group number 3. So I'll just pause here for a minute and say that if there's anyone from LNID in the room here, I'm sure they're getting really excited and they think, awesome, we can pass the title of the district with the most manure over to RID. But I do need to defend RID in this, in that RID's water, RID has very good irrigation water. So their nitrate levels in their irrigation water are often too low for isotope analysis. But they really wanted to participate in this project. So we told them to go and seek out sites that they thought might be the worst in their district. And I'll also remind you that the results from this project here are not meant to be interpreted as general water quality across the whole district. We have another project that does that, but this was just specifically sites were chosen to be able to evaluate this isotope analysis part. So I think that this shows that RID did a really good job of picking sites. They happen to be geographically very close to feedlots in their district. So that's what we're seeing represented in this data here. So. And then the final group is, let me see here. There we go. Group number 4 was a really small group. It was associated with fertilizer, inorganic fertilizer. So we just had less than 1% of samples from the BRID and the SMRID that kind of fell out in that organic fertilizer. So I'll just quickly speak to this, that we were kind of excited because we could see the land use of we could see the land use of the districts being reflected in this data. So SMRID and BRID, I think are known to be more crop production type districts. Even though they do have some livestock, they're not really intensive livestock districts. So it's kind of nice that we see, well, it's not nice that we see, but it's nice that we see that the nutrients in their canals are more associated with soil nitrogen. And then LNID and RID, for the reasons I just discussed, tend to be more livestock-focused districts. So it's not really a surprise that the nutrients we would see in their canals are more associated with the manure. I'll just move on now to another way to look at objective number 2, which is the specific sources. I'll just check my time there. The specific sources and sinks. Pardon? Oh, I thought someone said something. So I have time, 5 minutes, yeah. So looking at the specific sources and sinks, again, this is kind of a bit of a messy graph, but it's isotopes. So we see the ratio of 15N on the bottom and 18O along the y-axis, and then the bubbles that you see are the ratios that we would see from those general sources. So the green bubbles are atmospheric deposition. We see some fertilizer sources there. The blue is soil nitrate, and the orange is manure. So, and then if you can see the legend over in the corner here, the sources and sinks that are demonstrated on this graph are, the circles are canal water, the triangles are manure, the squares are surface runoff, and the the crosses are tile drainage. So similar to what we saw in the previous graph for BRID and SMRID, which are the kitty corner districts there, we see that most of their canal water, which are the circles, are kind of falling out in this soil nitrogen or soil nitrate bubbles, which is similar to what we saw previously. And kind of as a nice check or kind of a confidence boost, it's nice to see that our manure samples, which are the triangles, they are usually always falling out in the manure bubble, which is good because your manure-derived nitrate your manure samples should have manure-derived nitrate in them. And then also interesting, the squares and the crosses, which are the drainage water, they're usually falling out in the manure bubbles too. So that indicates that the nitrate that's in that runoff water is mostly manure derived. So I just said all those there. So yeah, moving into objective 3, which is basically how do we use this data to inform management decisions. So just we can recommend and adopt nutrient management practices that reduce the loading from the sources of concern. So as I said previously, if you're finding that your nutrients are coming from manure, then you need to target manure management practices in your district. And the same thing if you have soil-derived nitrate, you need to address soil erosion. As far as evaluating the stable isotope analysis as a tool for assessing nutrients, just to summarize, it was successful for carbon and for nitrate. As I said, the phosphorus concentrations just were not high enough, so we couldn't use it for phosphorus. So in summary, nitrate in irrigation canals is manure or soil-derived. Surface and subsurface drainage waters have manure and soil-derived nitrate, so likely best to avoid contributions into canals. And the other thing I really wanted to emphasize is the value of research partnerships. I'm not an expert on isotopes. I don't think the irrigation districts are expert on isotopes, but we were able to partner with the University of Calgary, who are experts on that, and then we were all able to use our skills and our resources to come up with a really great project. So yeah, this project was funded by CAP funding from the Risk Mitigation Irrigation Conveyance Works program. The 4 participating irrigation districts kicked in matching funding to be able to access that grant. The University of Calgary, I say very graciously, gave a lot of in-kind time of one of their grad students to help us with the analysis and interpretation of this data. And that's it. So maybe there's time for a question or two. Oh, Colin, I told you not to ask me hard isotope questions.
Is this on? Yeah. So thank you, Janelle. I just wanted to ask, is isotopic analysis a costly thing, and can it be done year-round?
Yes, it is expensive. And can it be done year-round? Like you mean, is the lab available year-round?
Yeah.
Or?
Yes.
You would have to, the university, well, I don't know if there's other places that do isotopic analysis, but if you were to partner with the U of C, they're obviously a university, so they are not a service lab, so you would need to have kind of a research partnership with them or have the project kind of set up with them. I think their lab runs year-round, so they could receive samples year-round. So.
Okay, thank you.